body defenses. So think ahead. All right, so at the end of last class, I was talking about the different categories that are part of body defenses. Um, there were, let me get a much smaller font here. We talked about general, also called innate. These are ones that aren't looking for a very specific thing. They're not looking for the novel coronavirus. They're just looking for something that doesn't look right. Or they might even be even more general than that, like, just your skin or mucus making it hard for things to get into you. In the analogy, general defenses would be like locking your door so people can't get in, or putting a fence around the property, or putting a moat around your castle with alligators. It doesn't matter who's trying to come in, it's just kind of keeping it out. The other side is the adaptive or specific defenses. And these are antigen specific. These are the ones that are going to be looking for and targeting very specifically shaped things. We talked about antigens when we talked about blood typing. This is what would be looking for specifically the novel coronavirus. Like, again, after we develop some vaccine, you will have lymphocytes in your body that are primed to recognize that and take it out so it'll never make you sick again. So we are going to look at both of these, and there are also some ways they interact with each other that we're going to see. So let's start with general, and then we'll get into the adaptive. Okay, so let's see, general defenses. So let's start with, and of these, there's everything. Yeah, humans like to categorize things and put things in boxes, and we just kind of, there's all these categories here. External. So external, these are things that are trying to kind of harden the defenses so things never even get in your body. So this is like thinking about your skin. Keratin. You have this really hard protein that makes the scales that make your skin actually pretty impenetrable. It's the same stuff it makes the horn of a rhinoceros, is also making this like tough coating on the outside of your skin. Sebum, which is that kind of oily stuff, which is released by the sebaceous glands. It keeps your hair supple and your skin kind of moist, but it's also antimicrobial. So you have that antimicrobial stuff going on there. Mucus, things that aren't your dry skin have kind of that sticky mucus. That sticky mucus both has just kind of the flypapery kind of thing, but it has what we call lysozyme in it. Let me spell it correctly. So lysozyme is an antimicrobial compound that's made by your, it's made by your body to put into all sorts of secretions. It's in your tears. It's in your saliva. It's in your mucus. That's, kind of all, coming out of your nose. And this actually breaks down the bacterial wall of yeah, it breaks down the cell walls of bacteria. So it's something that's antibacterial. So that mucus is not just sticky, it's also kind of toxic for a bacteria. So there's that. So what else? There's acidity. So lysozyme is something that's antimicrobial. And I think, I don't know if I ever mentioned this story, like the way, it's another one of these serendipity stories of these, somebody was looking at bacteria on a microscope slide and had some snot drip out of his nose onto the slide and noticed that something in his snot was lysing the bacteria on the slide. And it was like, oh my God, what's going on here? And they followed up and found that there was this compound that was in the mucus, that was actually helping lice break the bacteria. So acidity, a lot of the bacteria that you don't like don't grow so well in low pH environments. So like the sebum, in addition to being antimicrobial, is also kind of low pH, makes it less hospitable for bacteria to grow on your skin. Acidity in the vaginal canal, there's like the friendly bacteria, like the lactobacillus that are there, create the lactic acid, which makes it low pH, which is inhospitable for like Candida albicans, the yeast, to grow. So it's not like there's not the yeast around, but they don't really take off because it's low pH and they can't grow very well. It's why women often will get yeast infections after they've taken antibiotics. If you take antibiotics for some infection, you're killing off the bacteria that you were trying to kill, but you're also killing off all sorts of bacteria, including your friendly lactobacillus in the vaginal canal that were keeping it low pH. And now that they are no longer doing that, now the little Candida albicans can start flourishing, and all of a sudden you've got a yeast infection. So this idea that low pH is another thing that keeps populations of bacteria down, or in this case, yeasts down that you don't want growing. There's also the acidity in your stomach, right? The low pH, super low pH. So this is like, on skin, also in stomach. The stomach is around pH 2. So as soon as you swallow things, pretty much things die in there. Or not as soon as, but as they get mixed up in the acid. Once we get to the lower parts of the digestive tract, it's loaded with bacteria, but there are no bacteria in the stomach itself. All right, so these are kind of external things that are trying to keep stuff from even forever crossing into the inside of your body. The next step is all the things that are waiting on the other side, assuming things do get into your body. So these are general defenses still, but internal. And there's going to be a lot of them. So let's start with phagocytosis. So phagocytosis, eating cells. Right. It's like there's some, don't want this guy in there. So you have some phagocyte comes in and envelops it. And that's phagocytosis. There are a few different cells that can do this. One of the types of white blood cells that is important for this are neutrophils. And I should mention, we're saying general defenses. So how do they know what to eat? A lot of these general defenses rely on particular kind of carbohydrates, for instance, that would only be found on a bacterial cell. So it would be, so that's not specific. I'm not looking for some specific thing. I just see something that I know can't be one of my own body cells. Like again, if I walked into the classroom and I saw like some weird insectoid creature sitting at a desk with weird scales over and like eyes all over its body, it's like, I know that's not a normal student that belongs in this class. It just is so not what a normal student would look like. So I can go and like go get my laser gun and blow it away or whatever. So that is a lot of these general defenses. There are these things called toll-like receptors that recognize things that just should not be there. So neutrophils, these are kind of like these frontline cells that go in where there's some infection happening. They actually leave the bloodstream and cross across the vessel wall. They call it diapedesis, where you can just ooze out of the vessel and into the tissue spaces. Injured cells actually release chemical messengers that tell the body like there's a problem here. Your bone marrow is gonna like totally start upping production of neutrophils. So more neutrophils are flowing in your bloodstream and they're leaving out of the bloodstream into the tissues. Again, that pus, when you have some infection, a lot of it is like dead and dying neutrophils along with all of the other, tissue and bacteria and stuff. These tend to eat like, I think is it they have a limited thing of like 25 bacteria or something before they get full of toxic things and die themselves. So these are not like they're not in it for the long haul. They are kind of, you center the front line, they kill and eat stuff, and then they die themselves. There's another kind of phagocyte that is more long-lived, the macrophages. So these are more long-lived. These guys can live for a long time. They can eat hundreds and hundreds of things and not die. These can act as antigen presenting cells. What that means is they'll eat something and then they'll take a little piece of it, what it looks like, and present it to the specific immune system and say, look what I just ate. This is what it looks like, and start creating clone armies that are going to fight this thing. Whereas, neutrophils just eat, and then die. The macrophages are kind of more like, again, I think of them more as like the officers versus like the infantry or something. They go and like, look what I found, and let's go report and like mobilize more forces. Some macrophages are wandering. They just go all over the place, just moving through the tissue spaces, all through your connective tissues. Some are fixed. Like there are specific populations, the Kupffer cells just live in your liver. Their job is just always hanging out in the liver and, filtering your blood there. Other ones have specific, like the Langerhans cells are moving through the layers of your epidermis, catching things. So some macrophages kind of have particular beats that they kind of do. Other ones are just, in general, they just go wherever. Again, they eat things, they present what they've eaten to other parts of the immune system to set up specific defenses. So I talked about sometimes these guys know what to eat just because it doesn't look right. Another way they know what to eat is because there are parts of your immune system that can put special markers on cells that target them for phagocytosis. So there's this term called opsonization. It means making tasty. So we're going to talk about different things that can opsonize a target cell. We'll see like this complement cascade will do that. Antibodies do that. So opsonization. So sometimes the macrophages or the neutrophils, I should say neutrophils and macrophages are kind of under phagocytosis. So sometimes you'll have something basically like put little handles on, not on him, on him. I'll have like some target here. And some little like handles are going to get put on there that is going to make it even easier for the phagocytes to recognize and eat it. So opsonization, making tasty, like, put little handles on. So we'll get back to this big time when we talk about complement and we talk about antibodies. So the verb is to opsonize. So we'll talk about it opsonizes its target, which means it's going to make it easier for the phagocytes to recognize it and eat it. So if you are some little bacteria hanging out in the body, you don't want to get opsonized, because that means pretty much the next thing that's going to happen is these big white blood cells are going to come and gobble you up. All right. So phagocytosis. And again, in this case, we are talking about general, because sometimes things get phagocytized just because they have markers that make them, it's clear they're not human cells. Sometimes, though, they are targeted for phagocytosis because they've been opsonized, and we'll see that opsonization can actually happen as part of specific defenses. Part of what antibody binding is going to do is actually opsonize the target. All right. So next, more general defenses. So one way you can take out a target is by eating it. Right, so those are the phagocytosis. But another way you can take it out is by poking holes in the membrane so it loses its ability to regulate itself in it. So we also have NK, natural killer cells. And these guys kind of come in 9 a.m. And they basically, think about them having like some kind of a dagger. And they just poke little holes. This is like, he's coming in and poking little holes all over this thing. So it's not actually using a dagger. It's actually using these proteins called perforins. So these are basically, like, if this is, let's say this is the membrane of this guy. Maybe I should make it red. These perforins are these little proteins that the natural killer cell will start inserting in the membrane of the target cell. And you insert them and they actually start assembling kind of like staves of a barrel, and they become ultimately a tunnel that is now a hole in the wall of the target cell. So there's a few of them. They assemble together and now there's a hole. And now this cell is all full of little holes and it's gonna not be okay anymore. So natural killer cells, again, they are recognizing things that don't look right, or cells. Often this kind of thing happens when you have cells that are even your own cells, if they're like full of viruses or something and you have to take them out. We're going to talk about, again, we talked about last time, I'll say again, your immune system is not just about killing, foreign cells. It's also about taking out your own cells if they have gone cancerous and they're dividing out of control, or if they're so riddled with bacteria that, or I should say viruses, that, they're a lost cause and we just need to kind of take them out of the system before they like open up and spill viruses out to the rest of the body. So natural killer cells, we're going to meet other killer cells when we get to the specific defenses, the killer T cells that are doing a similar thing like this, but in response to infection by a specific pathogen. These natural killer cells are, again, part of general defenses. So natural killer cells, more general defenses. So complement. So this, I always dislike the name of this one because it's so generic. It's actually an actual thing. These are plasma proteins. This is like 20 plasma proteins. That activate in a cascade. So kind of like, blood clotting, like, you start this process and then there are these different things and you finally have, like in blood clotting, in the end, you catalyze fibrinogen to fibrin and you have a blood clot. In complement, what happens is you end up having membrane attack complex. So ultimately results in, let me do the typing. So ultimately results in 1, membrane attack complex. This is very similar to the perforins. It's basically holes in the target membrane. Opsonizes target. So it's basically putting those little handles on it. So now that target is going to be more easily recognized and eaten by the phagocytes in your body. And 3, intensifies inflammation. Now we're going to talk about inflammation, what it is and why it's useful in a little bit. The reason why complement is called complement is because it complements different aspects of your immune system. Complement can be triggered just because things don't look right, kind of as one of the general defenses. That's why I'm talking about it in under general defenses. Sometimes just because something has the wrong markers, it has markers that would only be found on a bacterial cell, this complement cascade is going to get activated. All of a sudden, the target's going to start having holes in its membrane. So this is holes in target's membrane. All of a sudden, the target is got all these little handles for the phagocytes to grab onto. And we're going to see why inflammation is also going to help with the defense here. Complement is also going to be a critical part of specific defenses. Part of, antibodies will actually activate this whole complement cascade. So when we get to like, why are antibodies bad news for the target? It's because they fix complement. They make this whole complement cascade happen. So I am sitting here and some antibody recognized me. Antibody is kind of like a little sticky wicket thing, but then all of a sudden complement starts happening, and all of a sudden holes start opening up all over my body, and all of a sudden the phagocytes see me and try to eat me, and it's like, oh my God, this is bad news. So complement can be activated just because things don't look right as part of general defenses, but will also be playing an important role in specific defenses, which is how they get their name complement. They complement different aspects of the immune system. All right, complement. Let's talk about more general defenses. Interferon. So interferons, these are basically kind of a virus attack warning. So does anybody not know the basics of what a virus is?
Could use a little review.
Okay, so viruses are weird because they're not really live. They can't replicate on their own. They're just basically some genetic material, DNA or RNA, and some little protein coat. And so they're not alive. It's just like some molecules sitting there. If they were just sitting on a table, they would just sit there forever. But if they bind, they usually have some way that they bind to a cell. And once that happens, they inject the genetic material into the cell, which then hijacks the cell's own machinery, the ribosomes and all that. And they have the cell start using the genetic material to make new viruses, replicating the viral genetic material and repackaging it. So basically they hijack the cell's machinery to create new viruses. So they don't have the ability to duplicate themselves, but through this kind of like, it's like this weird system of locking in and hijacking, they can actually make the cell start replicating the genetic material, recreating the protein coat, repackaging them, and then as the cell fills up with new little viruses, it finally lyses and spills out the new viruses all over the place. So they are there now to affect even other cells. So when a cell is under viral attack, it releases interferons, which then tell other cells that they should harden their defenses. So basically, When other cells see these interferons, then they know, like, let's like start like slowing down our all of our like protein synthesis and stuff like that. Let's make it harder for something like this to hijack the system and start replicating inside. So that's the basic idea of interferons. Are they tell cells that one of their neighbors is under attack and that they should, save yourself. It's too late for me, but save yourself. That's the basic idea of interferons. When a cell is under attack by viruses, it releases these. This is detected by neighboring cells, and they will take steps to make themselves less vulnerable to viral attack. Yeah, some of these viruses, for not being alive, they're pretty freaking clever. Like, there's some viruses, I was like reading about ones where, because if you just, if all of these cells were infected with viruses already, then if this cell lysed and just spilled its viruses out, but all of the cells that were already lysed, were already filled with viruses, then it's like, you're not really spreading your infection very well if you're just spilling out more viruses around a whole bunch of cells already filled with viruses. So some of these viruses actually make the cells make these little microtubule like kind of structures that come out like this. So when the viruses are spilled out, when the cell is finally completely filled and falls apart, these little extensions of the other infected cells act like these little like pole vaulting things that keep pole vaulting the virus out to the very edge of the boundary of infected cells. So it's now where there are uninfected cells. All right, so viruses can get, and they mutate really, really freaking fast. And most of them aren't killing people. The soil is full of bacteria. Your skin is full of viruses. Your skin uses viruses as mutual symbionts to help protect you from bad bacteria, right? Because viruses actually often just attack bacteria, bacteriophages, and that's actually could be a good thing for your epidermal cells to harbor, some viruses that are going to help take out bacteria. So viruses aren't necessarily, they're everywhere. They do all sorts of things. They have all sorts of different targets. And, some can be your friends and some of them obviously are not your friends. So interferons, more general defenses. Whoops. Fever. So this one is kind of cool. This idea that, when you're sick, you run a fever. It's not because things are malfunctioning and like homeostasis is going off. You actually have signaling molecules released by white blood cells that go talk to your hypothalamus and reset your temperature. People talk about endogenous pyrogens. This is basically signaling molecules released by your white blood cells that talk to your hypothalamus to increase your overall temperature. Endogenous, created within pyrogens, create fire. In general, there are different hypotheses about why this is useful. I'm not sure if I've ever read a compelling argument of slam dunk, this is why fever makes sense. But it's clear that it is something that your body's doing on purpose. So, in general, if you have a mild fever, it used to be like try to knock fevers down. Now, if you have a mild fever, they like, your body is doing it on purpose. It's probably helping for some way or another. So just let it run. Obviously, if it gets too high, if it's over, I forget what some number like 103 or something, where if it starts getting too high, it can start causing problems and other things can start getting, it can get dangerous. So then if fever gets really high, you do try to knock it down. But if it's a low grade fever, now the thinking is, it's actually your body is doing it on purpose. So it's probably good to let it go. Your white blood cells release all sorts of, we're going to talk about this idea of like interleukins and cytokines, interleukin between leukocytes, cytokines, just signaling molecules between cells. There are so many signaling molecules involved in the body defenses, where different cells are talking to each other, where the different cells are talking to other parts of your body. The white blood cells also release things that talk to your liver to try to pull iron out of your bloodstream, which makes it harder for like different pathogens to thrive. So we're not going to get into it. In fact, I would say these stories are constantly evolving. I mean, it's every week, like Science Magazine, there's always some new insight into like, oh, that's going on in the immune system. The immune system is so complicated and there's so many different interlocking mechanisms going on. So fever, which other one do I have here? Oh, I know what I got to talk about. Inflammation is the last thing for the general defenses.
Can you explain pyrogens? Like, just sum it up for me.
Yeah, white blood cells that are in the middle of fighting an infection will release these signaling molecules, which we call pyrogens, that talk to your hypothalamus. And the hypothalamus, if you remember, that was what set your body temperature. So it sets your resting body temperature higher than the normal, like 96, whatever it is normally, 97. So, and in different people's there's a range of normal body temperatures. It's not exactly, but once it gets over 100, it's probably in fever range. Sure. So general defenses, inflammation. Inflame, just from its name, is red and fiery. There's like 4 hallmarks of inflammation, and we'll talk about each of them. They talk about redness. That's why it's like inflamed. Heat. Area tends to get hot. Swelling. Area tends to swell up. And pain. It tends to get painful. So these are like the 4 hallmarks of inflammation. Let's talk about more on a physiological level how those come about and why it's a useful thing. So things that happen, vasodilation. More blood is coming into the region and the vessels get leaky. So 2 things are happening here. By having vasodilation, bringing more blood into the region, vessels getting leaky, this, allows, white blood cells to easily get into the tissue, to fight the good fight. This also allows antibodies and complement to get into the tissue easily to fight the fight. The fluid that's leaking out of the vessels is called exudate. That has the antibodies, has the complement. Another plasma protein that is in this exudate that is going into the tissue is fibrinogen. Which we've met already. Why would it make sense to have fibrinogen going out into this area where the infection is happening?
To stop it from spreading?
Exactly. So it's going to basically physically wall off the area so the fight is contained and the infection is contained and doesn't just start spreading systemically all over the place. So the fibrinogen, which turns to fibrin, is going to create kind of a wall that is going to, it's like think of an abscess. It's like kind of the infection is raging, but it's confined in a particular area. So the fibrinogen helps confine things. The redness, the heat, the swelling, those are all kind of coming from this, right? Vasodilation, more blood is getting red. More blood is also making it hot. Getting more exudate, the fluid going into the tissue is causing the swelling. The pain comes both from like the pressure of, on the little pain receptors, but there's also particular signaling molecules that are being released that activate the pain receptors as well. Pain, again, is helping the area heal by telling you, like, stop doing that. Leave it alone. Let it get a chance to heal. It's like, doc, it hurts when I do that. It's like, stop doing that. So, in general, pain is your friend to allow the area kind of a little bit of respite so it can heal. Obviously, chronic pain is a whole different thing. And we don't really have time to talk about that, but chronic pain gets very tricky to deal with. But all right, do I want to say anything else about this? All right, so that's basically, so inflammation, in general, it's happening for a good reason. Obviously, if it gets out of control, it can actually cause problems. But, it, at least initially, it starts to bring more blood into the area, bring in things that are going to help deal with whatever's happening. Okay, what's time? Let's move into the adaptive defenses now. So I'm going to start by saying how the two main branches that we're going to talk about, but we're going to start with the commonalities. So all of your adaptive defenses have three main things in common. They are always antigen specific. So we're out of this general defenses now. We are going to be looking specifically for the coronavirus or for the polio virus or for whatever. And again, I talked about the basic idea of antigen when we were talking about blood typing. So you've got a basic idea. It's basically just some shape on the outside of the target that you can recognize. Another thing that is a hallmark of the specific or adaptive defenses is they have memory. So once you have been exposed to something once, it takes a little while for your body to have a response, which is why you get sick and then you get better. But then you have memory of that. So if you ever encounter that same antigen again, the system can almost instantly like ramp up its defenses and you never get sick in the first place. And that's how vaccines work. We're going to talk about vaccines in just a few moments in more detail. But basically, The vaccine is about giving you memory, giving your immune system memory without hopefully making you sick in the first place. So then if you ever encounter that particular antigen, you will be able to take it out before it makes you sick. The last thing I'll say that's common is they're all systemic, meaning it doesn't matter where you encountered the pathogen, it's going to have this whole system activated all over your body. Like you might have first seen this pathogen in your toe, but then it shows up in your nose, and you're going to have, B cells and T cells and antibodies that will take it out there. So it's not about fighting some infection in some particular region of your body. Wherever you got exposed to something, it doesn't matter. It's going to create a defense that is going to cover your entire body. That's why we say it's systemic. So antigen specific, has memory, and systemic. So next, what we can do is talk about the different branches and how those branches are different. So these are commonalities. I'm gonna have to. Okay. Next, we're going to talk about the two branches of the specific immune system. There's going to be antibody mediated versus cell mediated. Antibody mediated is sometimes called humoral, which we'll talk about later. So let's talk about how these are different from each other. Just from their name, antibody mediated versus cell mediated, that's referring to the kind of primary mode of destroying the target. Here, antibodies are used to destroy target. And as we saw, antibodies are actually plasma proteins. They are like those immunoglobulins, gamma globulins. They're not cells. They're plasma proteins that have a way to bind and recognize and bind a specific antigen. And then we're going to talk later about why that takes out the target. But the antibodies are used to destroy the target for antibody mediated. And cell mediated, you basically have like killer cells take out the target. So these are going to be these killer T cells that are doing the same kind of deal that the natural killers were doing, inserting the perforins into the target cell and taking them out. Another big difference between these two, antibody mediated, this uses B lymphocytes. Where cell mediated uses T lymphocytes. Sometimes they just call these B cells and T cells. They get their name based on where they mature. The B lymphocytes mature in the bone marrow, so B for bone. The T lymphocytes mature in the thymus gland, which is up kind of over your heart. So T for thymus. So they're both lymphocytes. They're both specific immunity, but they mature different places and they're going to play different roles. We are going to continue looking at specific or adaptive immunity. And let's start by looking at antibody mediated. So the first thing, so we talked about, so let's antibody mediated. Obviously, we talked about antibodies being these little proteins that have this kind of variable region. So an antibody is just it's a protein. Has this variable region that can have different shapes that recognize, kind of recognize and binds the antigen. So the question then is, how do we come up with these? And it's not probably what you think. It was definitely not what I thought. I kind of figured, oh, it's kind of like, you mold it on there or something, or you kind of construct it because you know what you want it to fit. But that's not how it works. So first thing we're going to talk about is how you actually end up finding or making these things that are going to find and match some particular antigen. So first, we're going to go back into before you've ever encountered the antigen, your body has made B cells that have all sorts of receptors on them that fit all sorts of things. Right, so we have receptors that would match something like that, or be the mate to that, or be the mate to that, or be the mate to that. But these are all pre-made. These are all pre-made in circulation. So it's this kind of shotgun approach. You kind of randomly make B cells that have receptors that have all sorts of huge varieties. Then, meanwhile, okay, nasty dude. He's coming in to get you. And luckily, your white blood cell comes in. Here's my macrophage. Your macrophage comes in and eats this thing. Yum. And totally gobbles it up. And then the macrophage has this, we're going to talk about this thing, it's called MHC2. It can actually take a little bits of this thing and put it on its outside and presents it to other cells. Maybe it's just the horn. So this is bit of engulfed engulfed thing to present to the lymphocytes. So this for kind of going back over here, this guy, macrophage, comes in, eats this thing. Maybe it just regurgitates one little part, one little specific, easy to recognize part of this thing that it ate. Now it can go along and say, here's a little distinguishing, recognizable thing of this pathogen that I just ate. And then it goes and comes into this scene. So it says, all right, here's a little bit of this thing that I ate. Again, maybe it looks like a little horn or something. And it's like, does this B cell match? No. Does this B cell receptor match? No. Does this B cell match? No. Does this one? It's like, oh my God, it's like destiny. It was the chosen B cell. So this B cell is going to come, and its receptor is just the perfect match that binds that antigen that's being presented. And then there's going to be this whole thing they call like co-stimulation. So you really, 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 really don't want to activate the immune system unless you're really, really, really sure it's the right thing to do. If you activate the immune system to recognize and kill a certain antigen, but you accidentally have it target like something that's good in your body, then you've got autoimmune disease. All of a sudden you're killing your pancreatic islets and you can't make insulin anymore. Or you're targeting your synovial membranes and you've got rheumatoid arthritis. Or you're targeting your myelin sheaths and you've got multiple sclerosis. So you really got to make sure that you only turn this on if it really makes sense. Allergies happen when you turn this on, and it's actually not a dangerous thing. Like, it's like that pollen is not going to kill you, and yet I am freaking miserable. So allergies are kind of this hypersensitivity when it goes. So you want a strong immune system, but you really don't want a twitchy immune system. you want your immune system to like. So and this process is really complicated. There's all sorts of these interleukins, these between white blood cell messengers, back and forth, and handshaking requires actually T helper cells to actually happen. We'll talk about T helper cells, but T helper cells are necessary for both cell and antibody mediated immune system. It's part of the reason why AIDS is so, or HIV is so nasty. It's HIV takes out the T helper cells, which means that it is going to dismantle all aspects of your specific immune system. All right, so let's say that this B cell with this particular receptor is the perfect fit for this antigen that's being presented. The next step is we undergo what's called clonal selection. So we basically take the blueprint that is used to make this specific receptor shape, and we're going to create huge clone armies of cells that are going to churn out antibodies that all have that exact same shape. So once the match is made, oops, good God. We get what's called clonal selection. So we are going to make armies of cells that all have that exact same receptor. For antibody mediated, there are two cell families that get made. We're going to have plasma cells And these are basically antibody factories. They make something, I think, like 2,000 antibodies per second. So basically, they are spewing out into your body antibodies that recognize that exact same shape. They're making 2,000 per second. And you've got, like, when you have swollen lymph nodes, like when people are sick and they have swollen glands, those swollen glands are actually your lymph nodes swelling up because you've got these plasma cells dividing like crazy, churning out tons and tons of antibodies that are going to target whatever kind of thing you're fighting at the moment. So those swollen glands are actually due to the proliferation of plasma cells, making all these antibodies that go out, and we're going to talk in a few moments like why that is going to take out their target. So plasma cells, the other thing that's made are going to be memory cells. And the memory cells are just going to go get stored away basically. So if you ever see that same antigen again, your body will recognize it and isn't going to have to go through this whole process to start with. It'll have a quick, fast response. So in fact, let's talk about memory right now, just for a few moments. And if we do this very generic graph, like immune response, whatever that means, and days, 1, 2, 3, 4. Okay. And we do like, right here, like antigen arrives. First exposure, it takes a while for the immune system to ramp up. It's like this thing arrives, you start getting sick, and your immune system recognizes it, presents, you start making clone armies, you got all your immune system on, but then now you're fighting and then it goes away and you get better, right? So you get sick in the meantime because there's a lag between when the pathogen got into your body, had a chance to start taking over, and the immune system took a while to respond. On a second exposure, your memory cells say, I know that, and boom, you've got this really fast, strong response. And basically what happens is you never get sick in the first place because your body has been able to neutralize the thing before it ever had a chance to start replicating and taking over and making you sick. So this is what we call immunity. Immunity is this memory of the antigen. So if your body sees that antigen that it's been already seen, you don't get sick because you have this fast, strong response that takes it out before it has a chance to make you sick. And there are a couple of ways, and there's actually some different versions of immunity we should talk about. So active immunity, versus passive immunity. So active immunity, you've been exposed. You make memory cells. So if you have active immunity, your body, your lymphocytes have done their whole thing, made their clone armies, made memory cells, and your body is always going to be ready to go to nail this thing. You know, there's two ways you can end up with active immunity. You can get it because you actually got sick by the thing, right? In which case you've been exposed and you make memory cells. This is also how you make what vaccines are, though. Vaccines, rather than exposing the person to kind of the pathogenic version of the thing, you give them either an inactive. There's a bunch of different kinds of vaccines. You know, vaccines could use a killed version of the virus, for instance, so that it can't actually replicate. It could be the virus, but kind of a much less virulent form. So it doesn't really make you that sick, but it still has a shape. What's important is the shape. You can even just give them like a piece of the thing, as long as it's going to be enough to activate. So you can either use killed or non-virulent or even just parts and pieces. But as long as it has that particular shape that your immune system needs to recognize that particular thing, that's what's important. So, and if you can get the immune system to activate and create memory cells against that antigen, but do it without actually giving the person the disease in the first place, that's what a vaccine is. That's the whole beauty, is like you get the system to have memory, have this fast, strong response, but they never had to get sick in the first place because you kind of, you know, activated the immune system with this non-virulent form of the antigen. So does that make sense? So that is active immunity. You have memory. There's also a way if somebody needs instant immunity. Again, for instance, I think I might have told you guys, like when I was in grad school, we went on this big like diving trip down in Baja, and I came back with Hep A, and I was just deathly ill, and all my roommates had to get shots of gamma globulin. So it was basically the antibody against hepatitis A. And so they were instantly protected. Their body was full of antibodies against this virus, so they weren't going to actually get the disease from me. But it was passive immunity because you basically get antibodies, but you don't make memory. Right? So like 6 months later, after the antibodies had cleared out of their system, they were vulnerable again. Whereas in my body, my immune system actually went through the whole process, like I'll never catch that thing again. But so passive immunity, you do not have long-term protection. You get instant protection because you've got external antibodies coming in, but you don't get long-term protection. Active immunity, you have memory. One kind of cool thing, one cool thing about passive immunity is you have antibodies actually secreted in mother's milk. So like when a baby is nursing, the baby can actually get antibodies from the mom. So whatever kind of things the mom is protected from, she can confer to the baby through passive immunity. So the baby's system is protected as well, you know, and that gives the baby a chance to have their own immune system kind of come online and, you know, kind of get stronger. And in the meantime, has passive immunity from antibodies lent from the mom. So passive immunity can work that way as well. What's the time? All right, so.
What's happening with COVID-19? Because aren't they saying that you possibly can get it again?
Right. I mean, there's so many conflicting things, but they're also saying like we're not going to be done sheltering in place till we have like a good vaccine. So it's like, obviously I think people are still trying to figure it out. I mean, one of the problems with some viruses is that they change their outer coating really rapidly. And when we talk about like HIV, it happens so fast that it actually changes what it looks like before your immune system has had a chance to totally take it out, which is why you never get rid of it. But other things change more slowly. I suspect we're going to be able to have a vaccine. And I think it's way too early to read something like that and believe it's true.
Yeah. So would that mean that never mind.
I mean, I think it would mean that there's either somehow it's sliding under the radar of your immune system. It could be changing rapidly. Again, some viruses do change. Like, and again, it's why you can catch the cold or the flu every year.
Yeah.
Like within a year, both of those look sufficiently different from what they looked like the year before that if your body sees them again, it doesn't recognize it and it makes you sick again.
Right. Okay.
Right. I mean, the flu shots are basically working by looking at people getting sick in the other side of the world right now and assuming that that wave is going to hit the US in 4 or 5 months from now. So let's make a really quick vaccine now and then have everybody take that in the US before the virus makes landfall in the US. So, and hopefully they're going to be able to do something like that with COVID-19 at some point. All right, let's talk just a little more about antibodies. So how do antibodies destroy their target? So if you think about it, from what we were talking about, an antibody is just a little thing, like here's some target, and it's got some markers, and the antibody kind of comes in. It had like the little thing that grabbed on. So these little sticky wickets, so they don't look that threatening at first, right? They're like, oh, I'm so scared. This little thing just stuck on me. Like, why is that going to ultimately take out the target? What did we see happening in our blood lab?
There's no defense.
So in our blood lab, what was the effect of adding the antibodies to the blood cells?
Clumping?
That clumping, that agglutination. So we saw that by grabbing onto one and then grabbing onto another, they can get them all clumped up together, and that makes it easier for just the phagocytes to kind of come in and just deal with them all. So they all get stuck together. That's one way, agglutination, which we've seen already. Another important way is by activating that whole complement cascade. So if you remember complement, this is gonna, what are the 3 things we talked about for complement? Anybody remember? Have it in their notes.
Membrane attack complex.
So membrane attack complex. Exactly. All of a sudden, like, oh my, holy crap, there's holes showing up. What's another thing?
Inflammation.
Inflammation. There's much more kind of flow coming in, which means more antibodies, more phagocytes, more complement for that matter.
Opsonizes the target.
And opsonizing, adding in those little handles for the for the phagocytes. So all of a sudden I'm like sitting here and it's like this little sticky wickets on me. I'm like, oh, that's how annoying. But then all of a sudden it's like, oh my God, I'm getting these big holes and ulcers opening up all over me. And oh my God, all of a sudden now these big white blood cells are coming in and gonna eat me. So even though the antibody in and of itself seems doesn't seem quite as threatening, it actually calls in these big guns that start causing major damage to the target. Then the third part that how they work is something they call neutralization. And this is basically by kind of blocking important sites on the target. So, an analogy again would be like these little things start sticking on me and it's like, oh, big deal. But all of a sudden they're getting all in between my fingers. I can't use my hands. Oh my God, they're on my face. I can't see, so neutralization is when the antibodies start blocking important like binding sites on the target so they can't function properly anymore. Could keep a virus from binding and getting onto the cell it's trying to infect. Rattlesnake antivenom. If you ever get bit by like a diamondback rattlesnake and they give you a shot of antivenom for that, it's actually an antibody against the venom molecule that does this neutralization thing. It binds the venom molecule and blocks these important sites on the molecule that are responsible for breaking down your body tissue. So that neutralization, it's blocking important binding sites on the target so it can't do its job. So these are ways antibodies destroy targets. The last thing I'll mention before we move into the digestive system, we are going to come back to finish up cell-mediated, but I want to make sure we don't feel rushed. So let's finish antibodies. It'll be a nice kind of division point here. So types of antibodies.
Things like the or the natural killer does not involve antibodies, right?
Correct. Yeah. Natural killer cells are going to be using these things like toll-like receptors. They're basically looking for markers that would only be found like on a bacterial cell and not on a human cell, but in a more generic way, not looking for some specific antigen. And we will on Wednesday, we'll introduce the killer T cells, which are like natural killer cells, except they are specifically looking for an antigen. So antibodies, the mnemonic people use is MAJ. I don't know, the woman serving you coffee at the truck stop or something. Hey Madge, how's it going? Hey sweetie. So antibodies, they're also called immunoglobulins, also called gamma globulins. These are all synonyms. Also known as. So what we say is IgM, IgA, IgD, IgG, and IgE. So MAJ is just the way to remember what are the 5 different kinds of antibodies. Not in any particular order other than it's, the word that you can make with those letters. So of these, you should be aware of a few different ones. So IgM, this is released in initial exposure. Whereas if you're having you're having your memory cells responding because you're seeing something you've already seen, that's going to be mainly IgG. This is during subsequent responses to antigen. So the reason why this is useful is by looking at what kind of antibodies are in your body, you can get a sense, is this the first time you've ever seen it? Or is this actually a recurrence of something like, herpes? It's like, did you just catch this now and you're having your first outbreak? Or did you actually catch it a while ago, but you thought you had the flu or something and you didn't know? And this is just a recurrence of this, coming back. You can look, are they IgM antibodies or are they IgG antibodies to that virus? And you'll know whether or not, oop, I just caught it, versus, okay, I actually caught it a while ago, but I am now having a relapse of it. So people use this clinically to figure out whether or not something is an initial exposure or something that's coming up later. IgG, and the way I remember this is like, in Spanish, like primero, segundo, first, second, whatever. That's how I always remember these guys. IgA is also useful to know about. Secretory IgA. So this is like, in, your mucus, etc. So, we talked about the mucus being inhospitable because it's not just sticky, but it also had lysozyme. It's also got antibodies in it. So that's another part of your, defenses is in that sticky mucus, you're actually secreting antibodies that could actually recognize and take things out just as they land in the mucus. IgA is also the stuff that ends up in the mother's milk when I was talking about the passive immunity through, like, nursing. The other ones, don't worry about. IgE is often implicated in allergies, although I don't know so much about it. IgA is only found on specialized like lymphocytes. Don't worry about it. But you should have a basic sense of IgM, IgG, and IgA. So, any questions about all this? Just to go back really quick. We've just been talking about antibody mediated. We're going to talk about cell mediated on Wednesday, but it's still part of specific defenses. We talked about to design or to end up with antibodies that are looking at a specific shape. It's a little non-intuitive. It starts out, you pre-make the lymphocytes with receptors that can recognize almost anything. And then when the antigen presenting cell comes in, that is presenting the little piece of this thing it just ate, one of those hundreds of or tens of thousands or whatever, however many there are, is going to have a receptor that actually binds it and recognizes it. So you take that one and in this complicated process, undergo clonal selection. And in that clonal selection, you'll make plasma cells. These are types of B cells here because we're in our antibody-mediated immunity here. We'll make plasma cells, which are basically antibody factories, making the antibodies that are going to do all that stuff we just talked about. Then you're going to make memory cells, which you're going to squirrel away for a rainy day when you get exposed to that same antigen in the future. And again, that idea of immunity. Once you have memory cells, if you see the same thing, you have a really fast, strong response. So you get better with, or you take it out of commission before you ever get sick, which is nice. Are there any questions about any of this stuff? Okay.
What is a mast cell activation?
Oh, so good question. I don't have it here. So mast cells are basically cells that release histamine. So that's part of the inflammatory response. So mast cells, these are cells that are just one of the classic cells found in your connective tissue. They're all over the place. In the bloodstream, they're called basophils. Which are pretty rare, super rare. These things can get triggered to degranulate and release the histamine, which causes inflammation. Like, I am experiencing these guys right now. I have allergies to some of the grass pollen that's in my yard. And basically, I have an immune response where that grass pollen is triggering the degranulation of mast cells in my nasal cavities and my eyes, and I end up sneezing and have itchy eyes. So that's why if you have allergies, you take antihistamines, right? Antihistamines block the histamine, and so you block that inflammatory response that results from the histamine. So mast cells are part of the triggering of inflammation.